Bioluminescence is visible wavelength light that is produced from a biochemical reaction between an enzyme called luciferase and its substrate. This reaction can be found in nature in some fungi, beetles, marine life, and in fireflies. The enzymes that produce light have been identified and can be incorporated into mammalian cells using gene editing techniques. The most common luciferase is called firefly, though there are other commercially available luciferases, including Gaussian ranilla, nanoluc, ocoluc, and click beetle luciferase, to name a few. For most luciferases, a substrate will be injected into the animal a few minutes before imaging. This substrate will need to be administered at every imaging session to activate the bioluminescence reaction. The luciferase substrate kinetics can be affected by temperature, anesthesia, disease location, stage of disease, etc., so it's important to know when the signal strength will be the highest and most stable for your disease model. Many companies offer cells that have already been modified to express luciferase. You can also develop your own Luc cell lines using lentiviral particles. One advantage to Revity's luciferase cell lines is that the firefly luciferase has been modified to emit at longer wavelengths. This modification is advantageous for in vivo imaging because longer wavelength light will penetrate through tissue better. In life sciences research, bioluminescence imaging has been used in almost every major therapeutic area to track disease burden over time in living subjects and to monitor biodistribution of viral, genetic, and cellular therapies. Benefits to bioluminescence imaging include the ability to monitor disease progression longitudinally and perform non-invasive quantification at multiple time points. Major limitations include poor penetration of visible light through tissue and the need to genetically modify cells. Therefore, this technique is not used in humans and can only be applied to small animals like mice or for superficial applications. To image bioluminescent light, we use IVIS systems, which stand for In Vivo Imaging System. In the IVIS, you can image live animals, cells, and well plates and resected organs. The IVIS is a light-tight chamber with a sensitive camera to collect light, ports for routing gas anesthesia into the chamber, and a heated stage to help anesthetized animals maintain proper body temperature. When you collect these images, the 4 acquisition parameters the user needs to consider are field of view, exposure time, f-stop or aperture, and binning. The images will appear as a bioluminescence image overlaid onto a black and white photograph for anatomical reference. Some IVIS systems have integrated X-ray or CT hardware that allow for additional anatomical reference. Questions about animal handling and anesthesia should always be directed toward your in-house veterinary staff and clearly defined in your animal protocols. Additional questions about experiment design, image acquisition, and image analysis can be sent to your team of Revity application scientists.